Hello, all you beautiful people. Welcome to my first actual instructional video where I teach you how to go through a scientific paper. Any scientific paper. This is a fantastic introduction to folks who want to follow along in my journal club videos. Perhaps read the article ahead of time and then go through my video to see if they picked up on the same things. Now, if you're a seasoned scientist, you may not find this video that helpful, but this is for people who've never touched a scientific paper or have much experience. This video contains the set of instructions I wish I'd been given at the beginning of my PhD. Reading scientific papers was never intuitive for me, and I always struggled in the journal club during those first few years. Quick note to you all. I'm going to be honest, the screen recording of this section may make you a little motion sick. My iPad decided to make life difficult while I was recording and wouldn't let me zoom in or out properly or even scroll right at times. I did my best to edit everything out. The thing is, due to how many side gigs I'm balancing right now in the real world to pay my bills, yay capitalism, I have to sacrifice some quality to get these videos out in a reasonable time. I hope you'll forgive me for my little editing stumbles. Okay, now on to the real learning. I want to point out that watching this video once will not turn you into a master at reading these papers. You're going to have to read a couple of them before you get into the groove of science reading. And that's going to be frustrating to hear, but that's one of the unfortunate parts of just practice. Before you even touch the paper, see if you can find any alternative sources to give you more of the paper's background or context. I'm talking videos summarizing the data, or small articles. For instance, my paper on Leyland was written up in a blog and given a small summary article in the blood journal it was published in. You should read all of these alternative sources first, so you know the findings of the paper before you even start. This material will take way less time to read and digest than the actual academic paper, and you should take advantage of that. Once you've done that, then you can download or print the paper. I prefer printing. And then we can start delving into everything. Now, this is an example paper. I will be doing this paper for a journal club, but not today. Today is a how to read this. All right, so let me write this off to the side here. What you will need. The paper itself, I print this out. I like writing on the actual paper. If you're someone though who likes the online stuff, go for it. But I like the physical. The reason why I like the physical is because then it usually frees me up to have more monitors, which is another thing that you can't see here, but I do have. I like to have multiple ways to look up definitions, look up molecules. I just have all of that at my fingertips while I'm reading these. iPad, computer, iPhone, all free game for this. You will need this. You will not be able to just read a scientific paper without these. And this is true for pretty much anyone in the field if they really want to examine this. And then the last thing you will need, writing utensil. Be that a pen, a highlighter, whatever it is. The reason being is because if you're learning how to read papers, you're going to be looking back and forth between pages, and it's going to help you see parts in the paper that you will need to reference later. It'll make sense in just a little bit, trust me. So let me erase this really quick because we actually need the title. So step 1, read the title. This seems a little bit intuitive and a little bit too obvious, but we really like to just give you the main point of the paper. And if you need to, spend time trying to figure out what each of these components are. So for this example, we say Talin 1 is the principal platelet wrap effector of integrin activation. Now, if you're not in the field, none of that will make any sense. So the first thing you're going to do is figure out, like, go on Wikipedia. what is talin 1? And write that here. And you'll find that that is a signaling molecule. Is the principal platelet RAP1 effector. Okay, so what is platelet? If you were to look that up online, you would go, that is a cell type. All right. What is RAP1? That is another signaling molecule. And then you'll also go, what the heck is integrin? That is a receptor that helps plant cells bind. For cell binding. And I'm not going to go through the entire paper here, but this is what you should be going through for just the title. You need to pick out, okay, these are the main players. I should at least know generally what the main players are. And you may or may not be able to decipher everything about what the paper is yet, just from the title. But you are now primed to know who is who, what is what are we dealing with here. So we are dealing with signaling molecules in a cell type, and that's somehow related to cell binding. All right, next thing we're going to do, always read the abstract. If the abstract is well written, you should be able to get the main points of the entire paper here. In fact, if you're in too much of a hurry and you're trying to find resources for your own paper, you just look at, you don't actually go into the actual papers. You usually can just skim the abstracts and then you can use what you find in the abstract for your own research. Again, that's assuming if you're like on a tight deadline and you need to read through all these papers. For when you're going through it slowly like this, you're going to take the time to read through it. And this is, again, where you're going to be underlining and looking up and defining. So it's a RAS-related protein. That's what RAP1 stands for. Look it up. The editor and me couldn't help but note some of the word choices in this abstract, so you're going to see me do a little bit of marking up here. I say erudite language because there are better ways to say that. You are trying to get everything into the abstract all at once. Of the platelet signaling pathway that results in talin 1, again, we looked that up earlier, binding to the integrin beta cytoplasmic domain. And again, you're going through this, and it's going to be a lot of underlining, and there's going to be a lot of this, and there's going to be a lot of highlighting, and there's going to be a lot of notes. It's going to take a while. But if you spend the time now reading through all of the abstract as well and just knowing what are the main points, what are the main players again, well, you're going to be set up even more. I'm going to now move to the next section here, which you would think is introduction. You can read the introduction first. Me personally, though, I save the introduction until the end of this process after I've seen all the figures. So what I usually do is I go to the figures first. Hold on to your horses. There we go. So I usually want you to go straight into the figures because you should be able to look at each of these figures and it should tell you data, and at the very end of the figure, you should be able to then use all the data to make a conclusion, the same conclusion that they made. And so here they have a example of what the DNA looks like, where all of the parts are, and then they are looking at each of these individually. And if you don't know what these are, follow me down here. This is a fantastic guide, the figure legend. So if we're looking at what is figure A, we would know that that is a generation of, that is not the one I wanted to choose. that is a generation of mice, to RNA, And again, you're probably going to have to look this up if you're not familiar with it. It's going to be frustrating. It's going to be slow. Editing back here, this is also a good time to go check out the methods section if the figure legends just aren't cutting it for you. The thing is, the more you do this, the more you will become familiar with what all of these assays are, what does this involve, what are all of these molecular players here. So this is like, it becomes faster the more you're in the field. Okay, and then if we want to know what B is, well, we look at B. Oh, this is sequencing of the mutated region, yada, yada, yada. And then we can say, what the heck is E and G, da, da, da. And what I like to do is I will then write the same thing up there if I need. Sorry, I'm moving this around. But I will write that. So let's say this is E through G, impaired integrin activation. Now here's the thing, if you go through this figure and you still don't get it, or you're still struggling to figure out what exactly is happening, that's when you go into the, oops, sorry about that. That's when you go here into the results. And you can read through the results. Really, you just try to find the figures in here and you read around it. So this is your figure 1A, this is your figure 1C, figure 1D, and you are going to go, well, I'm having problems with figure 1A. So I'm going to underline everything underneath around figure 1A reference right here. Read that, look up what I don't understand, and then this will also tell you how to interpret if you can't do that yet from the actual figures. Ideally, you should just be able to look at the figures and interpret it the way that they do, but if you're struggling, this is where you find the explanation for the interpretation. If you're wondering why I'm not recommending reading the results all the way through first before the figures, I've always found just reading the results so overwhelming. The figures put everything into context, and if you can figure everything out from just the figures and visuals, then the reading of the results section becomes a lot less stressful. All right, so once you have that, I personally like to write summary of this figure. Usually there's one at its own, and in fact, you can find that they also have their own summary of the figure. I always write my own to make sure that I understand what the heck I just read. Then you're going to, you don't necessarily need to read the tables the first time around. I say tables for like the second or third time through, if I need it. And then we do the same thing with figure 2. We go, what's this? What's this? What's this? What's this? How do I read that? Same with this. If I don't understand that, I can go down to the explanations here. So this is all the figure legends. I understand what the A is. I read this, what's exactly happening. I still don't understand the interpretation. So I'm going to go back to where the the results talk about figure 2, which is how they set it up. I think it's all the way up here. No, I was too gung-ho. Figure 2. All right, so we're in the figure 2s here. So like, okay, we have figure 2A and whatnot. So now I can read through more of a walkthrough here and the explanation here. This will tell me the reasoning and what I should be interpreting. And again, you do that over and over and over and over. And when you get to where you can go through the tables, I go through the figures first before I touch the tables. Same deal. Although I really use the results section. And you're going to do that for the entire paper. Just keep going one by one, using all of your monitors, all of your note-taking to keep going. And of course, they always put things past the discussion, but keep scrolling until you've gotten all of the figures, Okay, and then if they have a summary at the end, this is amazing. Find summary. Figure. They may or may not have this depending on the type. Wow, I did not spell figure. There we go. All right. Sorry if this is glitching. I am using my iPad. All right. Then we go to the discussion. So this ideally, if it's written well, this will have a summary for us, what we just read, of all data. And it should have a why do we care. Okay. And this is actually the time to just read through and then go back to your figures if you need to. Did you get the same conclusions that they did? This is also when you start to ask, do I believe what they conclude based on their data? This is where you start to have people saying, I'm not sure they actually showed what they think they showed. Or you have people go, I think I am convinced. I think that they did a thorough examination of the paper or of the data. It is in the discussion section that they should be talking about future directions based on the data that they have. They should also be talking about the weaknesses of the paper. That is a good sign of a fantastically written paper if it also is able to say the models that we were using could not cover XYZ. So they should address weaknesses and future directions. Okay. This is why I have such a pretty handwriting. Okay, and now, this is when I go back to the beginning. I know this is all over the place, but now that you've actually gone through the data, now that you've actually gone through what everything is, now I read the introduction. Now this is where we go. What did I just read? What did I just do? Now, if you're wondering, when do we read methods? Well, we read them when you're not familiar with assay or experiment. So that's the first one. If they pull out a brand new experiment and you're like, what the heck just happened? Look at the methods. Usually in the papers of your field, you should know the general idea of a lot of the experiments. For example, I don't have to look at what did they have to do for Western blots unless I think data looks weird. For example, I've done Western blots enough times that I would be able to look at a Western blot, and if it looked off, I would go, what the heck did you do? And I would be able to look at their methods here, and if I see something wrong, or if I see something that they did differently, that would clue me in on to maybe why the Western blot looked wrong. If you're also curious as to maybe where get mice or a model animal. Again, that's where you're going to find that all in here. So I don't usually have to read the methods But when you're just starting out, I would say read the methods later, or if you really need it for context. All right, and that is how we read a paper. Expect to do 2 or 3 read-throughs of a paper, especially if you're going to do like a journal club on it. For the journal club videos, I'm reading these papers at least 3 times so that I'm able to try to walk you through them. So this is a good time to go through what you should be annotating for your graphs. And again, the more you do this, the less you'll have to annotate. But when you're first introduced to all of these types of concepts through graphs, you're going to need to annotate. So here I have a bar graph, but there's a couple different ways you can also show your data. There's also the, little violin plot where they show you the spread of the actual points this way. And they'll show where the median is. I can't show you everyone because there's just so many to cover, but these are the basics of annotation is going to be the same. So first off, you might have different color. Like for here, maybe let's say they used red over here. And then the other one, they used this light blue. Okay. Immediately start looking to where they define the colors and therefore the groups that you're going to be referencing. So this is group 1 and group 2. Let me add that. This standard deviation bar usually. This is important because sometimes people forget to change the colors, but they might still change the groups. So when you're going through a paper, just make sure that you understand who's who, because they might use one color set for one graph, and then they might be talking about the same groups, but they'll use different colors. It's annoying, but it's the reality that you might have to face every once in a while, more so for older papers. Okay, so that's your first thing you're going to do. The second one is that you're going to locate your X and Y axis. So here I'm just going to choose John A %, and then thrombin, concentration, let's just say it's molarity for whatever reason. Okay, so you have your X and your Y. What you're going to do, unless you know this off the top of your head, is you're going to define what each of these are. So in the case of John A %, I'm going to go off to the side here and redefine it for myself as how much integrant activation has occurred, so that I don't have to keep flipping back and forth to the results section and redefining it. You can even simplify it down even more, where you're just amount of activation. Okay, sorry if you can't read it, but you get the idea. So that's the first one, and then you want to do the same thing. So here you're going to go, this stimulates PAR receptor, or again, it depends on what you're looking at. But that's how you're going to go through all of your graphs when you're trying to learn it, because you're trying to understand, what are they trying to say? Are they actually saying that if I stimulate through this pathway, they'll do this or that? Again, that's the joys of trying to go through graphs. I might take that out. Okay, the next thing you're going to go through is the statistics. So you might see something like this, where they've done a statistical analysis between these two groups, and there we go. They've done statistical analysis between these two groups, and they might put something like NS, which just means not significant. There's no significant difference between, or mathematical difference between these two groups. Why I'm saying that is because all that stats is doing, at least what you'll usually run into, is how likely is it that the differences I see are due to chance. Right. So what NS means is that there may be differences, slight differences between the group, but that can be explained through chance. And then you're going to see what all scientists love, which are the stars. If you see a small star, that means it is significant. But if you see 2 stars, it's even more significant. And even 3 stars and 4 stars, it's even more significant. Oh, we love these little stars. Let me just draw all the hearts around them. The more stars, the more we love them. There is a whole discussion to be had about whether or not statistical significance actually equates biological significance, but that's beyond the scope of this video. Anyhow, these are the basics that you should be looking for as you're going through the data in your paper. Thank you so much for sticking around and watching the video, folks. If you'd like to ask for more how-to videos, you can contact me at my work email right up there, gettingthescienceright@gmail.com. And if you're interested in any of my services, well, you can also contact me at the same email, or you can check out my website. Anyway, thank you so much for your time, and have a great day, folks. Happy reading.